my group has been collaborating for nearly a decade now with Guy Ben-Ari and Philip Gamblin, who are at Symbiotica in Perth, University of Western Australia. They built this robotic arm that draws, called Mayart, that was controlled by neurons in my lab in Atlanta. This was the first embodiment of the cultured neurons that we grow on multi-electrode arrays. So we interface them to a computer and send the signals across the world to this arm that draws. Well, this Silent Barrage is the next generation of Mayart. So it's a new embodiment. It's got poles that represent the electrodes in the culture dish. The culture dish has an array of electrodes embedded in it, and each electrode is picking up signals from neurons in the culture dish in my lab right now. What's really cool about this is the closed loop. We built this closed loop system so that the video cameras that are in the ceiling are watching us, and when we move around, signals are sent back to the culture dish in Atlanta to stimulate those cells. The whole idea of Silent Barrage refers to the barrage of activity that the cells have when they're not getting any inputs. So if they're in sensory deprivation, they sort of go into this epilepsy mode of activity. And these waves of activity spread across the dish in a huge barrage. Of course, it doesn't make any sound if you're listening to the culture dish, but the irony of it is here in Dublin, you hear a lot of noise when these things have a barrage because all these robots move. So this has sort of caused a spin-off project in my lab that's a collaboration with Bob Gross's lab at Emory. They're working on trying to treat epilepsy in patients that have intractable epilepsy that can't be cured with drugs. They're facing the possibility of having part of their brain removed, a fairly big chunk of their brain removed, and we think they might prefer to have electrodes implanted in their brain. And these would deliver electrical stimulation very similar to what we're delivering to our culture dish here. We found in culture that this spreading electrical stimulation across the culture dish quiets the activity down and stops these barrages from happening. To say that this is like a living system it's actually not metaphorical, it's actually true. You know, these cells are alive. We have to work hard to keep them alive. We feed them the right chemicals, which are basically the components that are in the blood minus the blood cells. We keep them at the right temperature and the right humidity, and we have to really work hard to make sure that the brain of this exhibit here stays alive. So there's no doubt in my mind that it's alive, and this became very clear to us that there's some ethical implications. One time when Mayart was on exhibit and all of these people were watching it, and there was a live link to the scientist in my lab who was running the show from there, and he said, "Oh, excuse me a minute, it seems that the culture has died, I have to put a new culture in place." And everybody at the exhibit was like, "Oh my God, the culture has died, you know, what happened?" And this is a fairly common occurrence in my lab. If we don't take care of them carefully, they die. By working with the artists, I suppose that they continually push me to consider the ethical and the philosophical implications of what we're doing. A lot of scientists get the very narrow tunnel vision and proceed with their research without thinking too much about the big picture. And Symbiotica is very good at evoking these kind of questions, getting people, by putting stuff out there that's edgy, that's possibly controversial, they get people thinking about these things and pondering, what kind of science are we doing? Why are we doing this science? Why should we bother incorporating science into art? Should they be independent? We often think of these as two different cultures, but in fact, the artists and the scientists do their business in a very similar way. They're both coming up with creative ideas and then implementing them in some sort of a complicated thing that might be an experiment, or it could be an exhibit at a gallery. In this case, it's both. I would definitely like to continue these collaborations. I think they have been very fruitful. I think that would be really interesting to have some embodiment for the culture dishes that was more anthropomorphic. So I think that's the exciting frontier, is to move in the direction of things that can behave as more like people, maybe not as people, but more like people, to get people to think about semi-living robots. What does it mean to have a robot that's part alive, part living, wet, squishy neurons?